onto lecture 16, the at least final one I'm going to be recording for a little bit because I know you guys are still going through lecture 17 tomorrow. But for lecture 16, integration and regulation of metabolism. So this is going to bring together everything that we just talked about, talk more so about regulation, signal transduction pathways that we've kind of touched upon briefly in class. So let's go through it. Let's do it. So just central themes of metabolism. ATP is universal energy currency. We know this. Cells run on ATP, and it's generated through oxidation of sugars, carbohydrates, fatty acids, and even amino acids as well. And it's used in biosynthesis. And in catabolism, NAD plus is a major electron acceptor that will say, hey, give me all the energy that you're harnessing from catabolic reactions, and I'll take it and bring it to ETC, into the mitochondria. And then in reductive biosynthesis, NADPH, pH, not NADH, NADPH is a major electron donor. It says, hey, I have a lot of energy. Let's build some molecules in these anabolic reactions. Then we have small set of building blocks to construct biomolecules and break molecules down into. Biosynthetic and degradative pathways are almost always distinct, not exact opposites at key steps. So like the best example I can give is glycolysis and gluconeogenesis. We know steps 1, 3, and 9 we have to have bypass reactions, or step 10, 1, 3, 10, or whatever it is. But yeah, we have to have bypass reactions, and that allows us, that makes it so that these alternative pathways are not exact opposites. They're very similar though. And then the acetyl-CoA pyruvate spot between glycolysis, Krebs cycle, all these other places is a key junction where it's a unidirectional path in vertebrates, but in invertebrates, we know that they have the glyoxide cycle. It makes it not unidirectional. So something good to know. Now, moving on to more of like just general terms, factors affecting activity of enzymes. These are 10 factors. I mean, they're pretty self-explanatory, like extracellular signals may phosphorylate, dephosphorylate enzymes, which affects their activity, transcription of specific genes, mRNA degradation, mRNA translation on ribosomes, all this stuff. Like, you don't really need to get too specific. Just know that there are multiple ways. You could be asked to like list 5, and that's pretty much it. You just list them. They're pretty self-explanatory too, for the most part. Then organ specialization. So this is important because the main takeaway is that our organs have unique specific functions to keep our bodies running, and our liver is the one that mainly handles all the metabolic processes, right? And that's why metabolic syndrome, one of these like clinical diagnoses that's taking America by wave, is the fact that the liver is unable to process, the carbohydrates the fats and we end up forming adipose tissue because the liver is like, hey, I can't even break it down. I have to form adipose tissue. And so that's exactly what adipose tissue is it synthesizes, stores, and mobilizes triacylglycerols, right? They're not necessarily the ones that are regulating metabolism. They just are like, oh, we'll store everything as fatty acids and fat. And so adipose tissue ends up growing, and that's what we call fat and all that stuff. And then there is the brain. Obviously, the brain really derives from the glucose energy source. It will prioritize itself over the rest of the body. So if like glucose levels are going down, the brain will be the last one to use up the glucose, really, because that's the one thing it needs to always run on. And then it'll switch to ketone bodies. Cardiac, also, we know it uses ATP, uses calcium. It generates lactic acid too, to a certain extent. Same with skeletal muscle as well. We know the pancreas secretes insulin glucagon. It's almost like a glucose sensor inside of it, and it can tell the body the blood glucose concentrations. Then we have the portal vein, which carries nutrients from the intestine to the liver, and that's where the liver receives, okay, is it low blood glucose levels or high blood glucose levels based on the insulin glucagon that's in the portal vein itself. Then you have the small intestines, which absorbs the nutrients that we consume, the carbohydrates, the lipids, the amino acids. It absorbs it all through the small intestines, and it gets into the circulatory system by bypassing or passing through the epithelial cell layer. So the liver, talking more so about sugar metabolism, let's say we form glucose 6-phosphate, right? We have glucose 6-phosphate in the liver. There are a couple fates that can end up resulting. We know liver glycogen through glycogenesis is one way. We know glucose just exporting to the blood is another way, gluconeogenesis. It's the final bypass step that we need to undergo. We know oxidation as well, like just basic glycolysis will also work. Then we know acetyl-CoA as a lipid storage. It'll get broken down, and then acetyl-CoA will then be converted into a fatty acid through fatty acid synthase pathway. And then we also know it could be used as a pentose phosphate pathway to form our nucleotides and to form our NADPHs, our reducing cofactors for anabolic reactions. We know it does that too. So there's a bunch of fates. This is what I'm telling you, like having that iPad and that like doc or whatever of all the pathways integrated together will allow you to know specifically how all these pathways relate. So from the liver, now we're moving on to amino acid metabolism. So amino acids, we know that the first one is that they can be converted into proteins by building them up, putting them together, condensation reactions. That's in the liver. Number 2 is transport through the blood to other organs. So the amino acids could be converted into, like, for example, glutamate to glutamine. Then glutamine goes wherever. Or no, something like that. Or we know that in like skeletal muscle tissue, right? Number 2, that alanine gets converted into pyruvate. Or pyruvate gets converted into alanine, and then it goes into blood back to skeletal muscle tissue, right? As you can see here for that. Number 3 is the precursors, so like nucleotides, biological amines, hormones. That also can be as a result from these amino acids. Number 4 is deamination reactions, which allows us to get rid of the nitrogen and convert it into urea, which then get excreted in the kidneys through the urea cycle. And then the carbon skeletons can be used as TCA or generate glucose, glycogen, or lipids. Again, they're anaplerotic reactions, so they are gluconeogenic to a certain extent. Some of them, leucine and isoleucine, are pretty sure the ones that are only ketogenic exclusively. And then we have the ammonia, obviously I mentioned that. And then the last one is deamination of alanine for muscles. This is an example of like the transportation of amino acids in the blood and whatnot. So alanine gets transported. And so then we have alanine, pyruvate, glucose in the skeletal muscle, where basically we're going to generate a lot of alanine. It gets brought into the liver. Liver converts it back into pyruvate, from pyruvate into glucose, and then it gets back into the muscle and gets stored as glycogen for future use. So moving on to now lipid metabolism in the liver, we know that there are a couple fates for the liver. The liver stores the lipids. So for number 1, we can store them as just fatty acids or like just huge lipid molecules. Number 2 is that we can take these fatty acids and convert them into acetyl-CoA via beta oxidation. Then that's where 3, 4 come in, where it's oxidation. We also know that they can be converted into ketone bodies as well. Once we break them down to acetyl-CoAs, we can turn the acetyl-CoA into ketone bodies. And then from there, we have number 6, cholesterol, which is from acetyl-CoA to cholesterol. Then it forms our bile salts, which can be used in the digestive tract, or it can be used as steroids and hormones in the blood. And then the last, number 7 and number 8, the fatty acids could be converted to triacylglycerols or phospholipids, which get attached to plasma lipoproteins because they can't go in the blood by themselves. And this will get transported throughout the body, mainly towards adipose tissue as well. And the number 8 is direct transport in blood bound to albumin or as lipoproteins. Again, it doesn't have to convert into triacylglycerols or phospholipids. It could just be a straight up free fatty acid that goes into the blood and then attaches to albumin. It's never that the free fatty acid then starts going in the blood around the circulatory system. It's attached to a lipoprotein itself. Next is adipose tissues. We know that it stores and supplies fatty acids. We know that there's a difference between white and brown adipocytes. White is for usually the fuel storage and fuel mobilization, whereas brown adipocytes have thermogenesis. They generate heat. It's more prevalent in babies, and it's because we have the uncoupling protein from ETC, where it basically decouples the proton gradient from the ATP synthase, and it generates heat as a result. So then the next one is the brain, and we know the brain prefers glucose first. It's going to be the last organ to use glucose when we're starving, and during starvation, when glucose is unavailable, when it's completely gone, the brain will then use ketone bodies, and it requires a large amount of ATP to keep running your brain. And so the sodium-potassium pump uses a lot of ATP to maintain those different concentration gradients, and because of that differing ATP, because of the constant changes in memory potential, and we need the sodium-potassium pump to operate over time, and that's going to use up a lot of ATP. And again, this is a cool stat. 15% of all ATP generated by the body is used by the brain. Moving on to the next one, muscles. So we know muscles are used for contraction, light activity, rest. We obviously know fatty acids, glucose, ketone bodies are going to be used. But in heavy activity, we first use phosphocreatine. No, it is not it's different. It's not. Never mind. We're using phosphocreatine, which is just stored ATP. It's made from glycine, arginine, methionine, and creates cofactors Adamet. And then it's a reservoir of high-energy phosphates as well on top of ATP. And it also uses muscle glycogen. So really, when you're like lifting heavy at a short time span, when you're sprinting, you're going to be using these two energy storages. Whereas like if you're just walking, you're going to be using fatty acids, glucose, ketone bodies, the things that are much more easier to sustain. And again, there's a lot of sources for a reason, because if your muscles were to run out of energy so quickly and not have any other source, you're going to cramp up. Your cross bridges are going to literally tear apart, and your muscle fibers are going to rip, and then that's where you get an injury. Now talking about the Cori cycle, this is how we connect the skeletal muscle to the liver, where the muscle is going to burn through energy, and it's going to burn through our molecules, our substrates, and it's going to generate waste products, right? It's going to generate lactic acid. We need to get rid of the lactic acid at some point, and it does so through the liver. The liver is like almost like our cleanup organ, like it cleans everything up. So for this, the Cori cycle, what it is it allows the muscles to maintain proper levels of glucose. And the way it does so is by processing lactate, which is generated through anaerobic metabolism in muscle. So the muscle generates the lactate. Lactate goes into the blood and then gets into the liver through the portal vein, as we mentioned before. And once the lactate is in the liver, it's going to basically... an ATP is going to be used in the synthesis as it gets converted from lactate to glucose. And now that it's glucose, it'll go back into the muscle and get converted back into glycogen. But that's why we have the lactic acid buildup, but we have no more glycogen. The glycogen stores leave, and we no longer can actually use energy to fuel our muscles. So we have to let this time-dependent process that's much slower to convert it back to glycogen. Hopefully that makes sense. I was just not like rambling. The Cori cycle uses 4 ATP per cycle. I will mention it. You don't need to know the specific mechanism because I think this is just like a thing you have to memorize, but it uses 4 ATP per cycle. And after heavy exertion, increased O2 consumption replenishes the muscle glycogen, as I mentioned, and it replenishes your phosphocreatine levels as well. And yeah, here are the few. You should know this diagram as well. I know this is like the most memorization heavy class, but you need to memorize the fuels of the major organs. So you have liver, adipose tissue, brain, skeletal muscle that's working and resting, and then heart muscle as well. You need to know what fuel store they use, preferred fuel sources that are exported as well. That is just stuff that you should definitely know. And then metabolism, fed versus fasting states, right? So let's first talk about fasting because I feel like that's a lot easier because it's a deficit. So in fasting, we know that we don't have that much glucose, and glucagon is going to get secreted. Glucagon, again, is the hormone that tells the body, hey, we need glucose right now. We do not have enough in the blood. So glycogen is going to get broken down to glucose. It's like the first thing. And it's going to be sent out into the brain and other organs, right? That's the first thing. Then the next thing is that gluconeogenesis occurs. And we're talking about this in the liver, right? So gluconeogenesis will then be the next thing that occurs because it's a little bit slower, it takes more time, and it takes more energy. Whereas glycogenolysis is pretty easy, it doesn't take as much energy, and it's fast. So that's the gluconeogenesis will occur. Now, triacylglycerols are released from adipose tissue. So the adipose tissue also respond, are like, oh, low blood glucose levels, take some of the acetyl-CoAs. And so it's going to send it to the liver to produce ketone bodies when we're fasting, right? And the acetyl-CoAs get converted to ketone bodies. The ketone bodies then get into the blood and finally end up at the brain. And so then that's another source as well. Fourth. Number 5 is that proteins in the muscle are broken down. This is where we're starving for quite a while. We've been using ketone bodies for a bit, and essentially proteins can be used as gluconeogenesis or ketogenesis as well. And again, it's all these in the fasting state, we have a slow TCA cycle because we just don't have the glucose there. And so now moving conversely to the fed state, we have dietary fats, which can be directly sent to the muscle and heart to be used for energy. So pumping when we're really well fed, our muscles can build up and whatnot, and it's just overall positive because of the dietary fats. And these fats can also be sent to the liver to be turned into triacylglycerols via VLDLs, and then it's stored in adipose tissue. So when we eat too much, it can end up having a surplus of these triacylglycerols that end up expanding the size for quite some time, persistently, of the adipose tissue itself. Then we have insulin levels rise, which the pancreas secretes it, this hormone, and that tells the body, hey, we have high blood glucose levels, we need to get rid of it from the blood. And so liver uptakes glucose. So it's sent out to the brain, other organs. It's broken down to acetyl-CoA for lipid biogenesis. So if we want to make those fatty acid, triacylglycerols, cholesterol, we use the acetyl-CoA by breaking down the glucose into acetyl-CoA. That's what I was mentioning. If we have high glucose levels, we don't want to do gluconeogenesis, but we still generate acetyl-CoA, so then we have to go a different alternative route, and that's lipid biogenesis. And then lastly is that glucose can be converted into glycogen. And then in terms of proteins, we build proteins up when we're well fed, and it can be broken down and used for metabolism in certain instances as well. And then here's this diagram depicting all that. The pink rectangles where the sources of energy come from and supply the body. So this is where, we can't just be self-sufficient, not eat anything. We have to eat stuff, and that's what the pink rectangles are coming from when we're talking about fed state versus fasting states. And I just want to emphasize the liver is like the relay station. Most of our food passes through the liver itself before it gets processed and sent out throughout the whole body and exported. Then now we're going to talk about hormonal signaling. So it allows for coordinated regulation of metabolic pathways. So types of receptors are cell surface receptors and nuclear receptors. Essentially, cell surface receptors are like in the name, they're on the cell surface, on the plasma membrane. So these ones are essentially like peptides, things that cannot freely diffuse through the membrane. So as you can see, we can either have metabotropic or ionotropic. Ionotropic just means that we have an ion channel, allows ions to pass through. Metabotropic is a G-coupled protein receptor where a hormone binds onto it and it induces some internal signaling conformation change, and it's going to signal a secondary messenger cascade and so on and so forth, and then change gene expression or non-genomic pathways as well, interactions. I know I said a lot, but it's just review ion channels and G-protein coupled receptors. That's mainly what these cell surface receptors are. So again, the hormones bind to the extracellular site, and they usually don't go through, but they allow other molecules that are either already in the intracellular or from the extracellular matrix to go inside. And then metabotropic generate secondary messengers, which are signal-carrying molecules, which alter transcription or alter activity of pre-existing enzymes. Like PK. PK is a phosphokinase A. And we know PK can affect the circuit pump. It can affect some other proteins like, I'm trying to remember, some channels, ion channels on the plasma membrane. PK is not necessarily going into the nucleus, it's just interacting with pre-existing proteins. And then there's nuclear receptors where, let's say we have a steroid as our hormone, right? We don't need a channel or a cell surface receptor to bind to the hormone. The hormone, the steroid, can just pass through the membrane and then pass through the nuclear and then bind onto a nuclear receptor. And then once it's bound, it will directly alter transcription of a specific gene. It'll directly do so. So it doesn't need to have all these transporters, these sophisticated pathway. It just it's so easy for it to go through these two different membranes and then allow to change gene expression. That's why steroids are so effective at low doses. I've mentioned this so many times, but at this point I'll just beat the dead horse again. But essentially, glucagon and insulin. Glucagon is secreted by alpha cells of the pancreas. Insulin is secreted by beta cells. Glucagon tells the body we're low sugar. Insulin says we're high sugar. And by high and low, I mean blood glucose levels. So, glucagon promotes glucose generation in the blood because we need that. And it does so through gluconeogenesis or glycogen breakdown. And it decreases glucose utilization because we already have limited, so why would we want to use more? It's just telling the body, hey, we need to disperse all the glucose from the cells into the blood, so it increases the blood glucose levels. Insulin does the opposite, where it says we have to promote glucose uptake, we have to decrease blood glucose levels, and we do so by decreasing gluconeogenesis, increasing glycolysis, increasing glycogen, fatty acid, and tag synthesis as well, because fatty acid and tag synthesis are all derived from acetyl-CoA, and that means that in order to generate acetyl-CoA, we have to break down glucose. So glycolysis is also activated in this instance. And then epinephrine is secreted by the adrenal medulla in response to stress, and it stimulates glucagon secretion and inhibits insulin secretion. Why? Because when you're in a fight or flight state, you need to move, you need to run, fight, or flight. You need your muscles to be at peak optimal contraction efficiency, if you want to put it like that. But the epinephrine is simply just telling the body, hey, you're secreting all this glucose in the blood, and your muscles are able to contract more strongly at an instant, right? That's just a tangent, but that's what I'm trying to say is that it stimulates glucagon secretion, and all that I'm trying to say is that it leads to the breakdown of muscle and liver glycogen. It leads to the breakdown of glycogen in the liver and the muscle, where more glucose is in the blood for these other organs that are necessary, right? Whether it's in the muscle itself or whether it's in the brain or whether it's in some other compartment. And it stimulates glycolysis because we need that energy. We need to generate that ATP right away. And then talking more so about diabetes, there's type 1 and type 2. Type 2 is simply just insulin resistance where the cells do not respond to insulin. Whereas in type 1 diabetes, we destroy the pancreas, essentially the beta cells of the pancreas, and we no longer secrete insulin at all. So for type 1 diabetes, we just don't have insulin in general, right? Whereas type 2 diabetes, we do have insulin, but the normal amount of secretion doesn't stimulate enough of a response in the cells to uptake the glucose in the first place. So that's the difference between the two. So type 1 usually develops earlier in life because it's antibodies, your immune system destroying your pancreas beta cells. Whereas type 2 usually develops later in adulthood, and it's a sign of metabolic syndrome usually. I know diabetes and metabolic syndrome are different, but they often are conflated with each other. And this is just my own like background. You don't need to really know this. I'm just yapping. But it affects 27% of the population, 90% of all diabetes cases in the U.S. And in terms of metabolic deficits, it's elevated blood glucose levels. In both cases, cells cannot uptake blood glucose, overstimulation of gluconeogenesis in the liver. The body thinks it is constantly starving, and it leads to hyperglycemia, glucosuria, dehydration, and osmotic diuresis. So, and then it leads to an overproduction of ketone bodies because the body, again, thinks it's starving, so it leads to ketoacidosis. That's why there's this common term known as diabetic ketoacidosis. And the blood increases in terms of acidity, and because it increases in acidity, if you guys remember that bicarbonate buffer system that we have in our blood and how it is exchanged with the lungs, the end result that I'm trying to get at with diabetic ketoacidosis is patients will often cough. Diabetic patients will cough. Diabetic patients will often cough to get rid of the carbon dioxide by expelling it out. And then moving on to the signal transduction, amplification of signals, there are 5 different types. So there's G protein coupled receptors, there are receptor tyrosine kinases, and then there's a derivative of it called receptor guanylyl cyclases, which is not too common. Then there's gated ion channels, and then finally adhesion receptors like integrins. And these ones are more so for cardiac muscles. They're attached to troponin. They change contractility of the heart muscle cells. So that's a very niche one. But the features of all of these that I'm trying to get at is that there's specificity in terms of what is the stimulus that induces the amplification of it, and then there's desensitization and adaptation. And then lastly is integration, where 2 opposing signals can lead to a net integrated response as well. So a lot of these can converge, and there's almost winning stimulus or winning effect. So talking more so about GPCRs, we have a G protein coupled beta adrenergic receptor, which this is norepinephrine binding to beta adrenergic receptors. It induces this G protein coupled receptor. It's a trimeric protein. So there's the alpha subunit, beta subunit, gamma subunit, and essentially will split off. The alpha subunit goes on its own, and it will have a GTP bound to it. So the alpha subunit GTP goes on its own and activates adenylyl cyclase. Beta gamma does its own thing. But the G alpha subunit with the GTP will activate adenylyl cyclase. And then from there, it'll convert ATP into CAMP. Look at the structures of the two. It's pretty intuitive. The phosphate group forms a cyc ring, essentially, and CAMP. And then CAMP activates PK, or it activates some other pathway. It does a lot of variety of things. So, and once the PK is activated, it does a bunch of other things as well. I'm going really fast because this should pretty much be a review for a lot of you guys. And then this pathway right here, this diagram is just talking about the alpha subunit, how it turns off. So the alpha subunit has intrinsic ATP or GTPase activity, which just means that we can actually hydrolyze the GTP into GTP, and when it's hydrolyzed, it'll actually combine back with the beta and gamma subunit to form this inactivated state, and then the signal is turned off. So talking more so about cholera toxin, it's a specific type of toxin. Essentially, ADP ribosylation inactivates GTPase activity of G alpha subunit. And so what ends up happening is if it's ribosylated right here, this is what it's showing, if we add this nucleoside group to it, to our G alpha subunit, it will keep it constitutively active. It will always be on. When it is always on, you can never turn off the signal, and that leads to a constant signal amplification. It leads to very terrible effects like diarrhea, constant contraction of the smooth muscle, which means that usually that's what diarrhea forms in your intestinal system. Moving on to the next one, there's receptor tyrosine kinase, which is just an insulin example. You guys should know this pathway where we have insulin bind to this tyrosine dimer receptor, and what ends up happening is it phosphorylates itself. Once it's phosphorylated, it has enzymatic activity and will interact with IRS1. When it interacts with IRS1, the IRS1 protein will become phosphorylated from this receptor tyrosine kinase, and once it's phosphorylated, it'll interact with these, what are they called? They're not transcription factors. They're intermediate proteins, essentially, but GRB2, SOS, RAS. RAS is the G protein that we have here that GTP will bind onto and activate this RAF1 primary effector molecule. Once RAF1 is activated, it'll convert MEK and phosphorylate it. And once it's phosphorylated, it'll phosphorylate ERK, and ERK can go into the nucleus and then from there interact with a bunch of things and to basically change gene expression of it. That's essentially what it is. And then how insulin affects glycogen synthase. So we know here that glycogen synthase here's what it is. So we know GSK3, if you guys remember the regulatory steps, glycogen synthase is active when it's not phosphorylated. But GSK3 is a kinase, and when it's active, it'll phosphorylate our glycogen synthase, and when it's phosphorylated, it is now inactive. But this kinase interacts with PKB in this PIC. And again, IRS1, if you guys remember from this, IRS1 also is phosphorylated, and this is an alternative pathway. And PKB will be active. And when PKB is active, our kinase will become phosphorylated and it will no longer work. Because it no longer works, our glycogen synthase is stimulated to be active. So this is a way that insulin can actually activate our glycogen synthase in an indirect mechanism, in receptor tyrosine kinase mechanism. And then, this is the pathway that IRS1 is phosphorylated. It activates I call it PIC protein, but it's PI3K, so a kinase, which will basically phosphorylate our PIP2 into PIP3. PIP3 will activate with PKB, will interact with PKB, and essentially the phosphate groups will activate it, and the GLUT4 receptors are stimulated, and on top of that, it'll also inactivate GSK3. And then gated ion channels, this is like pretty self-explanatory. Essentially, a ligand will bind onto the ion channel, and then it'll open it up, and then ion can flow through. It's closed otherwise. If the ligand is not bound to the receptor, so like if it's acetylcholine receptors, if the acetylcholine is not bound, these ions cannot diffuse through because the pore is closed. And this is just a classic acetylcholine receptor. Honestly, like realistically, the things you have to know. Everything here is on the slide if you need a review, but most of it's pretty self-explanatory. And then nuclear receptors. So these are activated obviously by steroid hormones. We usually went through this. The steroid hormones themselves usually need to be carried by binding proteins in the bloodstream. They can't just freely travel, just like with fatty acids, with tags, with chylomicrons. You need chylomicron to grab onto the tags. And then with steroids, it's VLDL, LDL, HDL. And then these steroid hormones directly regulate gene expression. The receptor in the nucleus will form a complex with the steroid hormone and then bind onto the DNA itself. And the specific part of the DNA is called a hormone response element, HRE. And then the receptor guanylyl cyclase, it's the activation of the receptor leads to conversion of GTP to cGMP, and then it has its own downstream effects. Honestly, I wasn't even tested on this. I don't remember it. I'd never seen it in these advanced classes, so honestly, it's just know that it converts GTP to cGMP, and it's very similar to adenylyl cyclase, where it will convert AMP to ATP to cAMP. And then there's nitric oxide, which is an unusual signaling molecule. So nitric oxide is created by converting arginine to citrulline using nitric oxide synthase. And the result is that the oxygen replaces the amine, and we end up forming our nitric oxide. And the gas can easily diffuse through plasma membranes. And it can be generated and act upon the same cell, so as a secondary messenger. And it can also diffuse onto neighboring cells, almost acting as like a hormone. If we were talking specifically, I think it would be called a paracrine signaling, if I'm not mistaken. And then practice questions with answers, same thing here. That's pretty much the end of the slideshow. That was all the lectures I covered today. I don't even know how I did that in all pretty much one sitting, but yeah, I'm wishing you guys luck. Please come to the review session. I'm going to spend much more time explaining everything in slower detail, but obviously replay the videos, ask questions on EdSTEM. My goal was to mainly just use my slides as a guide to figure out, okay, what's the most important things I need to know, and then from there work and prioritize. And also just a note is that a lot of these slides, I put these red dots on. The red dots mean that they're important, that you should stick with them. They follow the format of Dr. Solomon's lectures. The red dots mean that, hey, you should probably spend more time on the slide and make sure you understand it. Whereas a slide like this, there's not really much. So you don't really need to know it, but prioritize this one last. So yeah, pretty much it. I'm wishing you guys luck. You guys got this. I believe in you.